So our last speaker will be Richard Roden, who will be talking about a different vaccine candidate, one that still hasn't been tested in people, and it's based upon the minor capsid protein L2.
Okay, so I'm going to try and do a whirlwind tour through the L2 vaccine field. We're obviously way behind where things are with the L1 virus-like particles. But I want to give a little bit of a historical perspective and give you a sense of the different approaches that are being taken with L2. So my disclosure statements, I have intellectual property relating to L2 that's been licensed by Sanofi, Pasteur, and GSK. So the fundamental question that people in the L2 vaccine field are really driving for is, can we develop an inexpensive pan-HPV preventive vaccine? Is that possible? Obviously, it's critical to protect against the two most important clades, the alpha-7 and alpha-9, that are the HPV-16 and HPV-18 related types. But there are also cancer-causing papillomaviruses in the alpha-5, alpha-6, and alpha-11 types. Species that we would like to be able to protect against. Obviously, if we're going to have the long-term goal of eliminating cervical cancer, we'd like to have broad protection against all of these types. I also want to mention that the potential for unmasking is that as we take out through L1 VLP vaccination, the two most important types, HPV-16 and HPV-18, that some of the what are currently relatively minor types will become much more important. This phenomenon has been called unmasking. There's also the theoretical possibility of type replacement, in which some kind of niche that has been left by the removal of HPV-16 and 18 is filled by the other minor types, although I personally am not a proponent of this idea. And then finally, we would like to be able to protect against other non-malignant HPV diseases, everything from genital warts to skin warts. So the story of L2 vaccines is actually a pretty long one. Studies in the early 1990s from actually numerous groups show that if you just made L2 protein in E. coli and vaccinated animal models with L2, they were well protected from experimental viral challenge with a variety of different animal papillomaviruses. And I want to point out that these models are the same models that were used to develop the L1 virus-like particle vaccines. I think part of the reason that the L2 was never really advanced from these studies is that the neutralizing antibody titers that were observed in these vaccinated animals were really quite low compared to the L1 virus-like particles. So although the L2 vaccines were shown to be very protective, obviously the L1 VLPs look very promising. One of the early pioneers in these animal papillomavirus studies was Severia Campo, and her group showed that if you vaccinate with either the full-length L2 in the green or just the N-terminus of L2 in the blue, you get excellent protection of cows from experimental viral challenge. But if you vaccinate with the C-terminus of L2, you didn't get any protection. And this strongly suggested that really the N-terminus of the L2 protein is what contains the protective epitopes. So a more recent effort has been from Martin Muller's group. So he's developing this same region of L2, but this time from HPV-16 L2. And he has 3 copies of pieces of the N-terminus of L2 fused to thioredoxin, and he's tried various pieces from a small peptide, 20 to 38, to a much longer 11 to 200 piece. So Martin is trying to develop this as a clinical candidate. we took a slightly different approach, although related. So it was obvious to us when we did an analysis of the sequences at the N-terminus of L2, this protective epitope region, there was a huge amount of sequence conservation. But although the sequence conservation was strong, it wasn't perfect. So we decided to take advantage of the fact that L2 was not a virus-like particle and that you could string together different L2s from different HPV types to form a multimeric complex. Again, similar to the approach that Martin Muller is taking, but in this case, we're using L2s from different HPV types. And so shown here is what we call the ×8 construct. It's the 11 to 88 region of L2 from 8 different papillomaviruses strung together as like a string of beads. I talked a little bit about the animal papillomavirus studies that have been done, but obviously we want to be able to have a model that you can look for protection against the human papillomaviruses. And John Schiller's lab developed the pseudovirus challenge model in the mouse. So in this model you can vaccinate the mice 2 or 3 times, and then after a certain period, then you can challenge the mice intravaginally with a papillomavirus that expresses a luciferase gene. And we use the luciferase gene because it produces light when infection occurs. So you can see on the right-hand side, the 2 mice, the right-hand mouse was not challenged, and the left-hand mouse was infected with the pseudovirus carrying the luciferase. So this is a very convenient model system to actually look at the cross-reactivity towards many different human papillomavirus types. So we use this model to try and look at the cross-protection seen with either the 2 licensed vaccines, Gardasil and Cervarix in the mouse model, or our new L2 multimer models. And this slide actually summarizes a huge amount of work, but essentially what we see is, here's the pointer. In this case for Gardasil in the red, that you get excellent protection against HPV 6 and 16, as you would expect in the mouse model, but protection against some of the other papillomavirus types is not as strong. Whereas with these L2 multimers, we saw very broad strong cross protection. And we essentially see a similar phenomenon with the Cervarix vaccine. Again, we saw very strong protection against, say, HPV6, but the protection against the, HPV 16, but the protection against other types was not as strong. But again, with the L2 vaccines, we're seeing very broad cross protection. So that's the mouse model. Neil Christensen's group developed another animal challenge model, which is actually a similar idea. But instead of packaging a luciferase-expressing plasmid inside of the papillomavirus genome, Neil's group uses the HPV16 capsid to package the cottontail rabbit papillomavirus genome. And this allows actually a disease endpoint as a readout in the rabbit model. And so you can do the same experiments, vaccinate the rabbits, and then challenge and see whether the rabbits get cottontail rabbit papillomavirus warts. And so this is some example studies done with, so this is a HPV16 challenge with either Cervarix or Gardasil. You get excellent protection in the rabbit model. Placebo, you see warts, and then also with the L2 vaccine, this one is being produced by Shanta Biotechnics, and I'll describe that a little bit more. You see excellent protection in the rabbit model, which has a disease endpoint. And this protection with the L2 vaccine is still completed at 1 year after vaccination. So Where are we with the L2 Multimer vaccine? Well, the bottom line is that Shanta Biotechnics, it's a company in Hyderabad, has been the one pioneering the manufacturing of the L2 vaccine. So they've been able to produce the vaccine at large quantities in bacteria and have developed a GMP process for manufacture. And right now, the product has just been vialed, and we hope to file with the FDA early in 2013 so we can begin phase 1 studies with this vaccine. So the animal studies have also shown that if you take the immune serum from a vaccinated animal and passively transfer it to a naive animal, you can get complete protection from experimental challenge. So just like with the L1 virus-like particle vaccines, the neutralizing antibodies induced by the L2 vaccine are sufficient to mediate protection. Although I point out this doesn't exclude a potential role for cell-mediated immunity. But it shows that at least that neutralizing antibodies are sufficient. But one major caveat is the neutralizing antibody titers induced by the L2 vaccine are between 30 and 100-fold lower than you would see with the L1 virus-like particle vaccine. Although I'd point out that these vaccines are still able to give excellent protection, even though the titers are much lower. So what are the key epitopes that the immune response is recognizing with these vaccines? Well, this is data from Kirill Kalnin at Sanofi. When he analyzed the responses of these rabbits to the L2 vaccines, he found that a particular epitope, which is at residue 17 to 36, which was originally defined by a neutralizing monoclonal antibody called RG1. So we tend to call it the RG1 epitope. It's right here. So this residue seemed to be, 17 to 36 regions seemed to be an important immunodominant epitope in the response to the L2 vaccines. So I mentioned earlier that one of the downsides of L2 vaccines is that the relatively low titers induced by vaccination with the linear L2 proteins as compared to L1 virus-like particle vaccines. So there's actually been quite a bit of effort to try and take advantage of the virus-like particle display to render L2 much more immunogenic. And one of the pioneers of this has been Bryce Chakarian at the University of New Mexico, and I thank him for this slide. Essentially, Bryce has been using virus-like particles derived from RNA bacteriophages, MS2 or PP7, as a scaffold with which to present the L2 epitope, this RG1 epitope I described earlier, with the goal of making it much more immunogenic. Obviously, the concern with having low titers of L2 antibodies is how long is the protection going to last if you have much lower titers than with the L1 virus-like particle vaccines. These bacteriophage can be produced abundantly in E. coli. They're very readily purified. And they're easily modified to insert these various epitopes. Interestingly, they also contain their own endogenous antigen, the actual the single-stranded RNA, so that acts as a nice adjuvant. And you can display these heterologous epitopes just like L2 on the surface. So this is an example of what he did. So in PP7, he actually fused the L2 epitope in the middle of the PP7 coat in one of the immunodominant surface epitopes. But in the MS2 particles, he displayed L2 actually at the N-terminus of the MS2 coat protein. So the important point of this is that the L2 epitope is tethered at both ends in this particle, whereas one end of the L2 is free in this particle. When he did vaccination studies with these two particle types, he found that when he displayed the HPV16 L2 epitope, that both particles induced robust responses against HPV16L2. But for the particles where both ends of the L2 were tethered, it was much less cross-reactive. But if the L2 was tethered just at one end, then he saw, as in the case of the MS2, he saw much more broad cross-reactivity. Importantly, using this virus-like particle display, he also saw very long-lived antibody responses out to 18 months in the mouse challenge models. The one downside of this virus-like particle approach that he's taken, though, is you get lots of antibodies to the MS2 itself. And Reinhard Kernbauer's group has taken another approach, essentially using the L1 virus-like particles as the scaffold to display L2. So he has these so-called RG1 VLPs. So these induce similar titers of HPV16 L1 virus-like particle antibody responses as the native particles, but they also have the advantages of inducing the cross-reactive L2 responses as well. So in summary, the seminal studies in the early 90s showed that the L2 monomer is broadly protective. There have been a couple of clinical studies over the years using either an L2 peptide, which was shown to be immunogenic but was never pursued, and there were some L2 fusion protein studies done by Cantab in the UK, but these were only in the therapeutic context. So we're actively pursuing the L2 multimer, hope to have it in the clinic very soon. Martin Muller has a similar approach that he's also pushing forward. And then there are several approaches of virus-like display of L2 epitopes, including in the bacteriophages, or actually AAV-L2, and also in the L1 virus-like particles themselves. So I'll stop there. Thank you.